Okay, that's fine. So just give me 1 second. Let me get this. Okay. I want us to, because people learn more when when you are more of a visual, and so I want to show you that. Okay. So let me look for, I think I should have issue. Okay, you can see my screen, right?
Yes, sir.
Okay. What I'm showing on my screen now is the structure of these two human angiotensin converting enzyme 2. So how many of us are in biological science? Like you've come across, you've worked on protein and the rest before. How many of us? Hello.
Can you repeat the question?
I said how many of us have actually worked with protein enzyme in the past?
I haven't worked with protein enzymes, sir.
If you haven't heard about it in class?
Well, I've heard about it, but I haven't worked on it.
Yeah, okay. How many of you have heard about protein? What is a protein? What is an enzyme?
So in summary, just kind of kicks up like a reaction, or like they're binding sites to it, where just in general kicks up body functions.
Okay, that's fine. Okay, let me just do a simple chemistry, just explanation. When you are talking about protein is a nutrient, let me put it that way. They are part of the food that we eat. Are you getting me? And apart from carbohydrates and lipids that we get from food, protein is, and in fact, if not the most important, let me say is one of the important nutrients that our body needs. Is one of the, in fact, if not the most, is one of the important nutrients that we know that makes up our body. Talking about your ear, your knees, everything in your system are made up of protein. Are you getting me? And the mechanical reactions or chemical reaction that takes place in our body, they require protein to function. Are you getting me? Now, this protein we are talking about, they are made up of different amino acids. Just like you have a building that is made up of different blocks, proteins are also made up of amino acids. Are you getting me? And there are different amino acids. We have arginine, leucine, valine, aspartate, aspartic acid, glycine, proline. If you look at this structure I'm showing to you now, this is a protein. Are you getting me? Now, if I should put the cursor on one, if you look at this now, this one give us, if you see, is 0.1 ASP. That ASP means aspartate. Different amino acid comes together to give protein. Just like a single block comes together to give us a whole building. Single amino acid comes together to give us a whole block. Let me show you something. I think that's where I should have started. Let me show you okay, protein. Protein building blocks. Can you see? This is just talking about the body composition. Wow. Can you see? Protein is a complex that is made up of amino acid. So when you eat meat, let me use meat for example, or fish. Fish and meat, they are very rich in protein. So when it comes to when you chew and you taste to your body, through digestion, the proteins are broken down into small pieces. Are you getting me? And the small pieces that they're broken down into is called amino acid. And these amino acids, they are the one that make up of your body protein, enzyme, hormones, and antibodies. All these things I'm talking about, they are protein. Even the hemoglobin that carries oxygen from in our body, they are made up of protein. And that is why anybody that has low level of protein in the body is at risk because of the importance of it. So this protein, this amino acid, they are being used for energy production. They are also important for glucose and fat. Do you know why amino acid protein are important? Everything that happens in human body happen because of enzymes. There are thousands of enzymes in our body, and all these enzymes I'm talking about, they are all protein. Every enzymes, even the one that metabolize glucose, carbohydrates, that metabolize fats, everything are made up of amino acid. They are made up of protein. So that protein is very, very important. So when you want to study, when you want to design drug towards a particular enzyme, is a protein, it is always good for you to have an understanding of the protein, of understanding of the enzyme. And all you need to know in this is to know that every enzyme you are going to work with when you are designing drugs are made up of amino acid. That's just the simple thing. And that is why I'm showing you this. If you look at this structure that I have here, this is the structure of ACE2, human angiotensin converting enzyme 2. This ACE2 is very important in our body. Apart from the fact that it accepts SARS-CoV-2 into the body, it is very, very good in regulating blood pressure and some other functions in our body. But when this enzyme is working, when this enzyme ACE2 is working very well, it accepts spike protein from getting entrance to the body. Let me just show you. I think I should let me show you a short YouTube video so that you guys can understand very well. So it's two receptor and SARS-CoV-2 entry. I just want you guys to just it won't take much time. This let me look for. Okay, this one is just 1 minute. So I want you guys to look at it. This is just an advert first. Now, this is the SARS protein now.
Dr. Edowu, I don't know if it's just me, but I can't hear the volume on there. I don't know if it's talking.
You can't hear. Okay. I think it's because let me just explain it. Let me explain it. Because I'm playing it. Okay, this is the structure. This is SARS-CoV-2 now. Are you getting me? This is structure of SARS-CoV-2. You can see that's how SARS-CoV-2 now. This thing that you are seeing, this is the ACE2 receptor on the surface of the lung, human lung. These are the ACE2. Can you see them? And we couldn't forget that this is angiotensin converting enzyme 2. These are the ones. Don't forget that the SARS-CoV-2 affect the human lungs, right? As soon as SARS-CoV-2 gain entrance into the body, it will interact with this ACE2. Are you getting me? Once it interacts with the ACE2, that is when it will gain access to the lung. You see, if there is no ACE2 there, SARS-CoV-2 is not going to get into the lung. But because of this ACE2, it's able to enter. So not only SARS-CoV-2, even the Middle East respiratory syndrome, coronavirus, and the SARS-CoV-1. All the 3 coronavirus make use of this receptor, this ACE2 receptor. And once it is there, the SARS-CoV-2 will interact with them. Once SARS-CoV-2 interact with them, it will not give the entrance for SARS-CoV-2 to get into the into the host cell. Did you get that now? That's just the simple concept that I want to show to you now. So, and this is the structure of the ACE2. Can you see that? This is the structure of the ACE2. And when ACE2 is functioning, it allows SARS-CoV-2 to gain entrance into the body, which is fine. Now, you as a scientist that want to design drug that will stop ACE2 SARS-CoV-2 from gaining entrance to the body, your own interest now is to look for a drug that will bind this enzyme ACE2 and stop ACE2 from accepting SARS-CoV-2 into the body. Am I clear?
Yes,
Hello?
sir.
I believe you understand the concept now.
Yes, sir.
If my presence at TSU gate allows me to bring in Temi to TSU, then if you don't want Temi to come to TSU, you can come and say, Dr. Du, don't let Temi come in. Because as soon as I'm not letting Temi to come in, Temi is not going to gain entrance into TSU. So the moment you are stopping SARS-CoV-2, that SARS-CoV-2, please don't allow, ACE2, don't allow SARS-CoV-2 to gain entrance into the lung. Then you are able to stop SARS-CoV-2. So now, how do you now do that? By looking for drug that will bind to ACE2, are you getting me? And stop it from accepting SARS-CoV-2 from gaining entrance into the system. That's that. So once you identify your target, I'm just using SARS-CoV-2 for example. If you're looking at other disease like HIV, there are also receptors. There are also proteins that allow HIV to perform its devastating effects in human. All you need to do is recognize your protein of interest. In this case, we are looking at ACE2 as protein of interest. In HIV, just recognize your protein of interest. Are you looking at HIV protease enzyme? Are you looking at integrase enzyme? Are you getting me? Just once you recognize your protein, fine. That is the first step when you want to design a drug. You must recognize your biological targets. And in this case, we are looking at ACE2. Apart from ACE2, in SARS-CoV-2, let me show you the replicating step of SARS-CoV-2. Let me show you so that I don't want your knowledge to be limited to SARS-CoV-2, but I want you to understand the basic concept. Replicating step. Or circle. The circle of SARS-CoV-2. No, I'm not using that. Okay, can you see my screen?
Yes, we can.
Yes.
Okay, now, so that you can understand, now, this is SARS-CoV-2. This is the replicating means from entrance to the point that is causing devastation. We are looking at the replication cycle. If you look at this place, this is SARS-CoV-2, right? This is ACE2. As soon as SARS-CoV-2 recognizes ACE2 and some other protein, they allow it to gain entrance. This is the human cell. All we have here is the human cell. This is the outside now. This is the human cell. This is the outside now, outside of the cell. So maybe someone sneezed or cough, and this SARS-CoV-2 is just moving in the air. As soon as somebody comes in contact with it, there are a lot of ACE2 in the surface of our nose, of our nostril. As soon as it comes in contact with it, the ACE2 will grant it access into the body. You see, as soon as it gains access into the cell, the genetic material, don't forget, virus cannot exist on their own. They cannot do anything on their own. If you put SARS-CoV-2 on your bench, on your desk, it will be the same way it is. Nothing will happen until it's able to get into a living host. That is when it can perform its function. And that's why in the field of virology, it is difficult to classify virus as a living thing or as a non-living thing, because it cannot do anything on its own. It has to look for a host before it can perform it. It can do its devastating function. So now, as soon as the ACE2 accepts it, you see, it gets into the body and it releases the genetic material. In the SARS-CoV-2 releases genetic material and gets to the body. And once it gets to the body, it will now start, arranging its genetic material through this process. So one of the enzymes, apart from ACE2, people also develop TMPRSS2, transmembrane protein receptor. People also look at cathepsin. People also look at the protease enzyme, the CL-Pro or the PL-Pro. People also look at the RNA-dependent RNA polymerase enzyme. Are you getting me? So as soon as this comes, it passes through this process. This single one that you are seeing here, by the time it passes through this process, it's going to produce hundreds of another. Only one came in, but by the time it comes here, it will replicate itself, give birth to many, and it will release thousands of it into the out, and this one will go and affect another cell. When you are developing drug for a particular disease, you have to understand the mechanism of that disease. And in this case, SARS-CoV-2 is what you are looking at. And so what are the enzymes that you target for you to be able to stop a disease? Are you getting me? And this is going to make me to give you this assignment. And this particular assignment, I want to have it. Please, I want to have it. And I'm going to give you that before the end of the day anyway. Now, once you identify your target, so apart from these two in this application I've shown to you, apart from these two, we can also develop drug for TMPRSS2, because it also accepts SARS-CoV-2 to gain entrance into the body. We can also target the CL-Pro, this protease enzyme. If you're able to stop this protease enzyme from working in SARS-CoV-2, definitely you are not going to get a mature SARS-CoV-2 that can cause damage to another cell. So some people develop their own drug and look to stop this enzyme. And what is it? This company, and I can't remember the name of it, I think JSK or so, or Mark, they discover a drug, Prenaval or so, against this particular drug, this particular protein, RNA-dependent RNA polymerase. How many of you have heard of remdesivir being used to treat SARS-CoV-2 before? Remdesivir.
Yes, sir.
Yes. What remdesivir does is to go and stop this protein from working. If you're able to stop any of this, if you're able to develop a drug, just one drug that is safe to stop any of this enzyme successfully, I'm telling you, you have already discovered a cure to SARS-CoV-2. And that is why everybody is trying to focus on this protein. Everybody is trying to focus on identifying a drug for each of these proteins. Everybody is trying to find a drug for each of these proteins, and that is what people are doing. So in your own drug design, as we are using ACE2 as an example, Temi can decide to use RDRP, and Nsendu can decide to use CL-Pro, PL-Pro, and Stephanie can decide to use cathepsin L. So everybody is trying to look at different enzymes on the replication of SARS-CoV-2, so that we can have a drug that can stop this. And if you look at the grouping I had, I try to give people, each of you guys, different protein of SARS-CoV-2, and that's what you are going to do. When you are developing a drug, when you want to develop a drug for a particular disease, just go and have understanding of the biochemical or replicating cycle of that disease, especially when it is an infectious disease. For example, when you are looking at tuberculosis, so I want you guys to go and do this is an assignment. Please take note of it. Please everybody should give me the replicating cycle. Replicating, just even you can go and download it if you like this, I don't mind. I just want to have an idea. Give me the replicating cycle of HIV1, and give me possible enzyme that you can target to design a drug to cure HIV. Should I repeat that statement? Hello?
Yes, sir.
Look for the replicating cycle of HIV, and under it, identify specific proteins that you can target for drug design. So in this SARS-CoV-2 now, you can target the spike protein on the SARS-CoV-2, you can target ACE2, you can target TMPRSS2, you can target cathepsin L, you can target CL-Pro, the RDRP enzyme. That's, for example, in this case. So please do the same for HIV. So even though our lead program this year is focusing on SARS-CoV-2, but I want you to be able to apply the knowledge to any disease. And apart from you understanding this, it will even help you in having understanding of disease diagnosis. You understand my point? So let's go back to the Chimera. So now, in this case now, this is the protein of our interest, right? And this is the protein of our interest, and it has different amino acids, right? So every protein, every enzyme, any protein, anywhere you see protein, hemoglobin, they are all made up of amino acids. And the second assignment I'm giving you, please, this assignment I'm going to get, I want them by Thursday next week. Give me the list of the first 20 amino acids. You have to know this. First 20 amino acids and their 3-letter code. Again, list of first 20 amino acids and their 3-letter code. For example, if you look at this point that I put here, this is leucine. The 3-letter code of leucine is LEU. Leucine is an amino acid. So it will give you the list of all the 20 amino acids and their 3-letter codes. Please take note of that. So that's 2 assignments now. So now, let me not give you the concept of drug design now. So we've identified our target now is ACE2. How do we identify a drug? Using computer-aided drug design to know if a drug is going to inhibit that protein. Are you getting me? Now, the concept is clear. Is clear. On every enzyme, there is an active site. On every enzyme, there is an active site. When a drug binds at the active site of a protein, if that drug is going to be an inhibitor, as soon as it binds on the protein, on this active site of the enzyme, it will definitely turn off the enzyme from performing its function. In this case, if you look at here, let me select these residues. You guys, you should get this software. Okay, now this is what this thing you see in is a drug. It is called MLN 4670. It is a drug. You can read about it. This drug has been shown I think it has even been approved by FDA to inhibit ACE2. It has been approved it is known to inhibit ACE2. So this is the active site. If you want to make this enzyme to stop working, this is the active site. If you want it to be working better, this is the active site. So any drug that you want to test on this protein must bind at this active site. And when it binds, there's going to be an adjustment. There's going to be a kind of adjustment of this whole structure. And this adjustment that happens here will determine whether this drug is going to be inhibited, this enzyme is going to be inhibited or it's going to be induced. Inhibited means it will stop the enzyme from doing its normal function. So in this case, if this drug binds to this enzyme and it inhibits this enzyme, it means you not allow this enzyme to accept SARS-CoV-2. But if this drug binds here and it induces, if the drug binds and it induces the enzyme, it means it will make the receptor ACE2 to accept more SARS-CoV-2. So inhibition and induction. Inhibition is to stop the protein. Induction is to make the protein to be more active. So for majority of drugs that you see on market, they are either inhibiting or inducing a particular protein in the body, and that is why they are able to work. For example, if you look, and you don't need to, if you look at the mechanism of action of Telenol that we did, just take any drug, put it on PubChem. PubChem will give you the mechanism of action. According to this, Telenol is there to go and inhibit COX-2 to stop the production of prostaglandin that bring about headache. Can you see how it works? And ritonavir or lopinavir is an HIV drug that when HIV patients take the drug will go and stop the HIV protease enzyme from working, so that there won't be much viral load in the patient's system. So that is how every drug that you see on market, that's how they work. They do not go there to perform magic. No, they have what is called a biological target, therapeutic target. So this drug, MLN 4670, the therapeutic target is the ACE2. And once it binds at this site, it stops this enzyme from performing its function. And by so doing, it will stop ACE2 from binding or accepting a spike protein from getting to the body. Did you get the concept now? Hello, everyone.
Yes, Dr. Dogo.
Yeah. So that is the concept of drug design. That is the concept that is being used by this big pharmaceutical industry, big Johnson & Johnson, being Moderna, being Pfizer, anyone you can think of. That is the basic concept of drug discovery. When you are looking at a disease, you must have a particular target. For example, if you take a drug now, let's say nobody knows about the drug, and you give it to a SARS-CoV-2 patient, and after some hour, the patient gets back normal. You see, the next thing that a scientist will ask you is, what is the target of this drug in the patient? What are the drug actually going to do in that patient for the patient to be okay? What they are asking you is, what is the biological target? Is it targeting the ACE2 in the body? Is the drug targeting the RNA-dependent the polymerase enzyme? Is it targeting the protease enzyme? So once you do that, that is the research that the scientists will be doing. What is the biological target of this drug? So when you have a drug that you are designing, you must have a particular target. So this drug, MLN, is targeting a particular enzyme, and that is the ACE2. So now, you want to design a drug now, and you are looking at, okay, let me look for a drug. Let me design my own drug, or let me look for a drug that is already on market that can stop this enzyme from working. How do I go about it as a drug designer? You know, how do I go about it? How am I going to come about it?
Good. Here's some
Hello?
information for Kenny.
Hello?
At what time would you test it?
Oh, Jessica, can you mute, please?
Please, are you guys following me? I want to hear from you. Are you understanding the concept?
Sir. Yes, sir.
Please, because we are going to work, I want us to cover a lot. We are going to
Why?
work on the software, do a lot of things. But I want you to understand the concept of drug design. When you want to design a drug, you have to be specific about a particular target. And to be sincere, there are cases whereby a single drug can target two things. As this drug is targeting is two, it's possible that it can target spike protein. It's possible it can target the protease, SARS-CoV-2 protease. It's possible it can target HIV protease. And that is what is called drug repurposing. You know, remdesivir was not meant for SARS-CoV-2. Are you aware of that? Hello?
No, I wasn't aware of that, Dr. Ro.
Remdesivir was not meant for, it was not meant for SARS-CoV-2. It was meant for Ebola virus in West Africa. But because Ebola virus also has what is called RNA-dependent RNA polymerase enzyme, which is also present on SARS-CoV-2. They now said, okay, let's go and look at the similarity in the structure of RNA polymerase enzyme of SARS-CoV-2 and look at that of Ebola. And they see there are some level of similarity. Then they assume if remdesivir can work for RNA dependent of Ebola, let's try it on SARS-CoV-2, maybe it's going to work. And it worked. That is what they call repurposing. The original purpose of that drug is for Ebola, but now it has been repurposed for SARS-CoV-2. So it is possible that a single drug can target different protein, either from a single disease or from a disease or from another disease. There is possible that you are going to get a drug that will target ACE2, target SARS-CoV-2 protease, and also target HIV protease. Is possible. So, but the concept is your drug must have a particular target. So now, let's now assume now we have a drug, we have drug A, drug B, and drug C, and we want to test. So let's assume now that paracetamol, Telenol, for example, Telenol, metformin, let's say Telenol and metformin. Let's assume that, let's see, can they stop this ACE2 from working? Are you getting me? Can they also stop ACE2 from working? We don't know, but you want to try. If you try it now using computer, we are not going to lab now, we are using the computer. How are we going to know? The best way to know is to make use of your computer-aided drug design. And one of the concepts of that is what is called molecular docking. You see, molecular docking is as this drug, this MLN, as this drug is standing, you know, the drug is already bind, right? At the active site of this protein. There is what is called a binding energy that exists between this drug and this protein. So let's assume that the binding energy between this drug and this protein is minus 10. Let's assume it's minus 10, right? Now, when you go and bring another drug, we are using Telenol and metformin, for example, in this case. So when you now use your computer, you will now take the drug and dock it at the active site of this same protein without this drug. So when we have this drug to this, we have minus 10. Let's go and take Telenol and put it here. What is the score we are going to get? Let's go and take metformin and put it here. What is the score we are going to get? But don't forget, this one is known. MLN is known. And the score is minus 10. So let's now assume that Telanol give us -7 and metformin give us -12. Let me ask you, and this one, the standard, the already known, give us -10. Which one do you think is likely to work as inhibitor of this drug between Telanol that has -7 and metformin that has -12?
-12. -12.
-12. Because this one is already known as inhibitor and it has -10. Definitely, any drug that you dock at this site, are you getting me, that has close to -10 or above -10 is likely to inhibit this enzyme. Do you understand the concept now?
Sir.
So how do you now do the, how do you perform the molecular docking now? That is the question we should ask ourselves. How do you perform the molecular docking? That is the question we are going to ask ourselves now. How do we perform the molecular docking? To perform the molecular docking, there are hundreds or let me say tens of softwares on the internet that you can use to do what is called molecular docking. The molecular docking will give you the binding of any drug at the active site of any enzyme or any protein. So that by the time you do that, you'll be able to compare the score of your drug to a known drug, like we just did between MLN and metformin and Telanol. So how do you now perform what is called molecular docking? So to perform molecular docking, let me have something. Let me have a Word document so that at least for you guys to be able to. So I'm coming, just 1 second. Oh. I think I've stopped sharing, right?
Yes, you're no longer sharing, sir.
Okay, can you see my screen now?
Yes, we can see it now.
So now, how do we do docking? That is the question we're asking ourselves now. How do we do the docking? Do your docking is simple. It is what? It is simple. It goes to computer-aided drug design. We are going to use Chimera software. Chimera software. Do you get me now? And now, when you want to use Chimera software, you need two things. The first thing you need is the protein of interest, right? If I need to get the protein, isn't it the enzyme? I can say protein, enzyme, or I can call it receptor. Okay. So anyone I say, I'm referring to the same thing, or receptor. Are you with me?
Yes, sir.
So I need to get this, and I need to prepare this. The second thing, I need to get my drugs, or I will call them ligand. Anyone. So these are the two things that I need to do molecular docking. These are the two things that I need for molecular docking: enzyme or protein or receptor, the drug or ligand. So what we are going to do now, the first thing now, let's get and prepare. So we need to go and obtain, or let's download and prepare your enzyme. That's the first thing to do. Download and prepare your enzyme. That is the first thing you do. And how do you now? So let's call the 4 with download and prepare your enzyme. How do we download the enzyme? That's the question we ask ourselves now. How are we going to download the enzyme? And to download your enzyme, simple. There is an NIH database called Protein Data Bank. Protein data bank is really available. Just go to Google or any browser you're using, protein data bank. It is called RCSB DB, protein data bank. Click on it. This is an NIH. This is an NIH database. Every protein all over the world are deposited in this database. Every protein that you can ever imagine of are on this database. And even if you do your own experiments in your lab, you can deposit here. It's allowed. Anybody can deposit here. Are you getting me? So this is where you are going to download the structure of your protein. Whether you are, whether you are looking for human ACE2, you are looking for monkey ACE2, any one that you are looking for, just come to this database. That is where you get it. And that is where we are. So the first thing in molecular docking is to download your protein and prepare your protein. Once you are done with that, the next thing is to download your ligand and prepare your ligand. After you have done the preparation of the two, you now bring them together and do molecular docking. Are you with me? Are you guys following?
Yes.
Sure.
Okay. Please, this is straightforward. I just use the pivot note. That's about 40 minutes to explain in detail, because I believe if you understand the concept, whether you are working with SARS-CoV-2, you'll be able to, you get it. That's why I just spent time. But now we are going to docking now, into the docking now. So now, once you are on this website, next thing to do is to search, is to type whatever you are looking for. So in this case, we are looking at human angiotensin converting enzyme 2, right? So type it. Just type it. As you are typing, you even be giving you a suggestion. Human angiotensin converting enzyme 2. Do you know why I put human? Because this enzyme is present in other animals. It's present in monkey, even in mice, in rats, in hamster, in anything we can talk of, and majority of mammals. Enzyme. So for you to be able to show you are downloading that of human, you have to put human angiotensin converting enzyme 2. So let me click, then click enter, or you click this. It's loading, and you will see the number of structure of this enzyme that is present on this website. Can you see? We have a lot. Can you see? And this is just one page out of 7,636 of this structure present on this website. Can you see? And that is it. So if you look at the first one, it's called inhibitor bound human angiotensin converting enzyme related carboxypeptidase. It's 2. Then don't forget, there is ACE1, human angiotensin converting enzyme 1. There is human angiotensin converting enzyme 2. The one you are interested with for COVID is human angiotensin converting enzyme 2. So when you are downloading any structure from protein data bank, you must be specific about the organism of your interest and about the protein you are looking for. Are you guys with me? So you have to be specific, because there are a lot of structure that can be similar. So if you look at this is one, right? And it says it even has inhibitor bound to it. So that's fine. If you are looking, then once you have this, another thing that you should be concerned about, there are a lot of mutations that can happen on protein. For example, when you are looking, for example, let's say you want to develop drug against wild type SARS-CoV-2 spike protein, you must be sure that you are downloading spike protein for SARS-CoV-2, not SARS-CoV-1. And because you are looking at wild type, non-mutated type, you must make sure that the one you are downloading has no mutation. So all those things must be considered. when you are choosing, because if you are not choosing the right protein here, whatever result you are getting is going to be like a false positive because you are not working with the right protein. So in this case now, we are interested in human H2, and this is human, and this is H2, right? We can say we are fine with it. So what you need to do is click on it. Now, this is the information about this protein. Can you see this is the protein? Can you see? This is the information. Classify is an aldolase enzyme. This is more of biochemistry. This organism is from Homo sapiens. It's from human. Expression system, use this organism to express it. Mutation is no. So if your work is not mutation type, then your mutation should be no. For example, if I want to work on delta variant of SARS-CoV-2 spike protein, delta variant spike protein, and let's assume, and I'm seeing mutation no, I should know that is not the right one for me because I'm interested, because the one of delta must have mutation. I must have knowledge of those mutations. So in this case, there's no mutation for this H2, fine. That's what you are looking for. That's what you want. Another thing, technical thing you consider when you are choosing this is you have to consider the method. You see that it is NMR or X-ray diffraction or X-ray crystallography. The resolution should be between 2.00 to 3.00 Armstrong. It's acceptable. Anything getting to 4 Armstrong is not a good one. The resolution is just talking about how the clarity of the protein. That's what that is talking about. Now, so this is the person that did this experiment in the lab. So you don't need, so they're just giving you information about the author of this manuscript. And now, this is giving you information about this structure that you are seeing here. Apart from H2 that you are seeing here, that is why I said you download and prepare your protein. Why I said you download and prepare your protein is because there are some structure that are not naturally part of the protein that will be on this protein. So when you are preparing them, you have to remove them. But you must know which one is the actual protein of your interest. If you come down here, this is chain, there are different chains. Chain A. The chain A is the original angiotensin converting enzyme 2 that we are looking for. The chain A. And this is the structure, is it? If you go down, there will be some other thing. You see, this is B. And B is just not known, it's not part of the protein. Let me give you an example of how this resolution works. You see this X-ray crystallography, they are taking, when they perform the experiment, they take the picture. Tell me?
Yes, sir.
Let me ask the ladies. Stephanie, are you there? Is Nosema there? Hello? It seems, guys, I'm just speaking to myself, right? Hello?
Yes.
How many do we have online?
Hello.
Okay. You know, when you are taking for picture, maybe with your friends, you take the picture. Apart from you taking the picture, that they are taking the picture for, there are probably some people working behind you that the picture is going to capture, right? They are not part of the picture, but they are just working behind and the picture captures them. They are not the main thing. So when they are taking this crystallography structure, it takes some structures to them. We call them non-standard structure. So when you get to the preparation, you remove all those things from your protein. And so from this, only chain A is the H2 that we are looking for. And it has 615 amino acid sequence. 615 amino acid sequence. So this chain B, when we get to the preparation, we have to delete chain B. We don't want chain B.
I'm sorry, you can have it now.
Hello. So we delete chain B, and if you look at chain C, it's not part of the H2. We delete chain C2. It's not part of what we were looking for. So chain D is not also what we are looking for. Only chain 1 is 2. Chain A rather. Let's go to, can you see? It's the only angiotensin converting enzyme 2. So if you look at other ones, they are disordered segments. We don't need them. The other segment you don't need all those ones. That segment you don't need all those ones. All this, even E, you don't need it. So we delete those ones. Now, apart from amino, there are some structures that are there. And if you look at this, headings, the title of this, it says inhibitor band. That means there are some inhibitor. There are some drug that is already bound to the enzyme already. So when you get to the preparation, you remove them. You remove, so, and that one in particular is this SS5. That is the inhibitor they are referring to. So this NAG, we delete it. Zinc. Another thing is, you have to read little about your protein. Zinc is part of ACE2, because ACE2 is a metalloenzyme. I don't know if you've heard about the word metalloenzyme. Metalloenzymes are enzymes that has metals. They can only function in the presence of metal. If there is no metal in their structure, they are not going to function very well. Example of them is ACE2 is part of them. I think hexokinase for glucose metabolism also is part of them, that makes use of magnesium. So they call it, so ACE2 is, this zinc structure is part of it. We are not going to, chlorine is not part of it. So we delete chlorine, and that's all. So once you're able to, you already identify what you want, the next thing is to, let's download. So before I download, let me create a folder on the desktop that I will put this. So create a folder on your desktop. Just let me call it LEAP. Let me call it LEAP, for example. Now, that's where I'm going to download my structure. So now let's go back to, now, go to the folder, go to download. Can you see? Just come here, come to this, go to download file. When you download file, if you download the FASTA sequence, it's just talking about amino acids. That's not what you want. What you want here is the PDB format. That is the format that can be opened on Chimera. PDB format. PDB format. Click on it, it will automatically download into your download. Can you see? So once you download it on your download, go to the folder and go and copy it. Let me copy this, Ctrl X, and let me go and put it in my folder, in the folder I just created now. Let me go to the folder. Where is the LEAP? Okay, LEAP, and let's paste it there. So we've downloaded our protein now. The next thing is to prepare our protein. We prepare our protein. Now, I'm going to send you a video of how to install Chimera. I think what I'm going to send, maybe I will put this, so I will download the software and put it in the Google link, then you can make use of it. Or no, I will make a video on how to download Chimera. So that's fine. So now, once you open Chimera, the next thing is to go to file. You have to open the protein. Just go to file, click open, file, open, and go, it will give you your drive C. So go to wherever you save, you save the download. I have it on desktop and I have it on LEAP. Okay. And this is the structure. Just double click on it. Can you see? This is what you downloaded. So you can move it around. You can move it around. When you are I usually advise people to use mouse when you are working with drug design, because using this won't make things easier for you. So I always advise people to use mouse. So you can zoom it in and out. You can rotate the structure. So what we have done now, based on this, we've downloaded our structure, our enzyme, our protein. The next thing is to prepare the enzyme. As it is now, we cannot do, as it is now, we can't do molecular docking on it. No. It's not going to give us the right thing that you want. So we've downloaded it. The next thing is to prepare this structure. I want you to take note of something. If you come to this select chain, we have chain A, B, C, D, E. And don't forget from PDB, only chain A is 2. Every other chain is what? They are not standard chain. So we need to delete, apart from chain, we need to delete every other chain apart from chain A. We refer to them as non-standard. So if you come to residues, let's go to residue, come to residues. These are the standard residues. Amino acid are referred to as standard residues. Every other ones, apart from zinc, which is part of this protein, are non-standard residues. So you remove them. Even water is there. What we need to do is to start removing them one after the other. So click on chain B. Can you see chain B here? Once you go, how do I do that? Go to select, come to chain, and click B. Can you see it highlights in green? The next thing is for us to delete. Come to action, atom/bond, delete. Can you see that thing is gone? Go again to select, come to chain, click on chain C. C is highlighted, right? Go back to action, atom/bond, delete. Go to chain again, go to B. It's highlighted, right? Let's delete action, atom/bond. If there's anything you don't get, let me know. So let's go. Which other chain is there apart from A that we need to delete? Chain E. Click on chain E. Action, delete. We have deleted all the chains. Apart from the chains, there are also some residues that is not. So you come to residues, select residues. We said chlorine, we don't need chlorine, right? Click on chlorine. Chlorine is highlighted. Go to action, delete. Let's go to the residues again. We don't need water. Water is not, we don't need water. Click on it. Action. Because the water molecule is too small, so we can't see it. Go back again. You can see the water and the chlorine is no longer there. NAG2 is not part of it. Click on it. Delete.
I have a question, Dr. Edowu.
Okay.
Yes, how do we determine what chain and residue are not needed?
Thank
or
you very
is
much.
that something we should know already?
Thank you. Yes, it's something you should know. You see, one thing that is always good to do is when you already have a particular target that you want to work with, it's good to go and read about that target. No, all proteins has amino acids. Apart from amino acid, what other thing is part of the protein? You understand my point now? For example, look at this H2. Apart from the amino acid on this H2, it also has zinc ion. Not all enzyme has metal. Majority of enzyme, only the amino acid, there is no any other metal again. But if you don't read about H2, you will not know that zinc is part of it. For example, if you look at spike, SARS-CoV-2 spike, like except the amino acid, there is no other thing. So then when that's one, you read about it, you know. When you go to your when you download your structure too, it's good to read what is there. If you look at this one, it says this chain A is the angiotensin enzyme that we are interested in. So I already know that chain A is the enzyme of my interest. Every other one, if you look at it, it says this other segment. It's not part of it. We don't need it. Can you see?
Okay, that makes sense. That makes sense now.
Can you see now? So, but even these ones, this is an inhibitor that is already on it before. So you need to delete. But the zinc now, that's why, you know, if you look at it, the zinc is similar to every other thing. We know that the zinc is part of the protein. But until you go and read about your protein, that is when you are able to know that there is zinc on it. Do not all ions are metalloenzyme. I said H2 is a metalloenzyme because they require a metal enzyme for their action. But majority of enzymes, only the amino acid is what you need. So if your enzyme does not require iron and you see zinc, chlorine, magnesium, you delete them because it's not part of them. We are only leaving zinc because zinc is part of these two. Do you get the concept now? So let's go back to Chimera. So now let's go to the residues. So just select residues. Now zinc is part of it, right? If you click it, I already clicked the zinc. Let me show you. You see the zinc at the center of the active site. You see the zinc highlighted there. Let me zoom it. Can you see the zinc?
Yes.
Now, so it's part of the enzyme. It's part of the enzyme. Now, the next thing is there's inhibitor. This inhibitor is already attached to it, right? Another question I'm expecting you to ask: how do you know the active site of an enzyme? How do we know the active site of an enzyme? There are different ways to know the active site of an enzyme. There are softwares that can predict active site of an enzyme for you. There are software, but I don't really depend on them. It's better you read and know them because the software may not be accurate. The software can predict different parts for you. Now, the best way to know the active site of an enzyme is to go and read about the enzyme. What are the amino acids at the active site of the enzyme? For example, if I read about this enzyme and the enzyme says, and let's say the enzyme, let's say the article says, the journal says, let's say this 515 up to 520 are the active site of a protein. All I need to do is to locate where is this 1515 to 120. This is 1515, right? So as you are putting the cursor there, it will tell you the amino acid and the residues number. This is tyrosine at number 515. Don't forget the whole sequence is 615. So this place is at 515, tyrosine 515. Are you getting me? So definitely at this point is the active site of my protein. If it says the active site is 590 to 600, I need to look for where is 590 and 600. Did you get it? So I know, then I will say, okay, this is where 590, 600 are. This is 595. Then this is the active site. So that is how you know it. That's one. Another thing is when you are downloading your protein from Protein Data Bank. Another thing is when an inhibitor is already bound to the protein, there is 95% that where the inhibitor is bound to is the active site of that protein. And for example, if you look at what we have here, it says, the title says inhibitor bound is 2. That means inhibitor, there's an inhibitor already bound to it. And that inhibitor is this. Is this compound. So that means anywhere you see this compound, that is the active site of the protein. And let's go back. Even though we know this is the active site, you can see the drug is already there, right? So this is the active site of H2. When you are downloading your structure, you are likely to be lucky to have an inhibitor, a drug that is already known to already bind at the active site. And if there is no drug bind at the active site, don't stress yourself. Just take the article, read about it, and it will tell you exactly the active site of your protein. Do you get my point? Am I clear? Any question before we proceed?
No question, sir.
Okay. But I believe you guys are, if there is any question, please feel free to let me know. I don't even for me to start from the beginning. I don't mind. My concern is for you, not for me to be talking, is for you to understand this whole thing. Okay, that's fine. So now, we know this is the active site of the protein, right? We all know that now. So let's delete this protein. Let's the drug. Let's delete the drug. So already highlighted, let's go to action, atom/bond. delete. Now it is deleted now. If you go to chain, there is nothing again, only chain A. If you go to residues, only zinc that we want. So we remove every non-standard residues from this protein. This is the one we want now. That's fine. Now, the last thing, for you to be able to do docking on a protein, you have to remove hydrogen from the whole structure. Should I repeat myself again? Anytime you are preparing a protein, a receptor, or an enzyme for molecular docking on Chimera, you remove hydrogen. So how do you remove hydrogen? Come to select, come to chemistry. Hydrogen is an element. Come to element and click on hydrogen. You know, I click on hydrogen. Can you see anything highlighted here?
No,
Nothing
sir.
is highlighted because hydrogen is already removed. If hydrogen is there, it will highlight hydrogen for us. I'm telling you, it definitely highlights hydrogen for us, but hydrogen is not included here. So we are fine. So once you have done this, what you need to do is to save your protein, the one you already prepared. How do you save? Come to file, go to save. Once you click on file, you have to save. You have three save rather. Save image, save PDB, save mol2. Anytime you are saving a protein, you save it as PDB, not as mol2 or image. No, as PDB, and you click on save PDB. When you click on save PDB, it will bring you to this window, and I want to save it. This is my lib, right? I want it to be in this folder. If you want to change it to another, maybe I want to save it in COVID, you click COVID and you save in COVID, but I want to save it in lib. So you have to be organized where you save your things when you are doing this work, because you are going to work with different enzymes, different diseases. So I put it under lib. So let's give it name. Let me call it prep prepared. I just put prep is 2. I purposely put prep because it has been prepared, right? So I call it prep is 2. So let me click save now. So just click save. It's automatically saved in your folder. Let me go to the folder and show you guys. This is the one we downloaded. This is the one we just prepared. So that one, the one we prepared, is ready for docking now. It's ready for what? For docking. So that's good. So let's go back to our Word document. So we have downloaded and prepared our enzyme. The next thing is to download and prepare your drug or its ligand. That's the next thing to do now. We have downloaded our drug. We have downloaded. We have downloaded and prepared our enzyme. The next thing is to go to the drug. So now, in this study now, we want to do a standard drug, a known drug, MLN 4670. We want to do Telanol. I want to do metformin, right? So we need to download the structure of the three of them. Are you getting me? Once you download the structure of the three of them, we will now dock them each. We want to see which one is going to give us close or higher value than MLN. Anyone that gives us close or higher value than MLN is going to be the one that is likely to inhibit ACE2, close or better than MLN. So now, how do you download MLN? And that is where, so once we are done with this, let me close this. We already saved it. Let's go back to our browser. So now, in this case now, if you want to download structure, which database do you think we should go? Hello? Hello?
I'm not sure.
Yeah, you can go to ChemSpider. You can go to PubChem. You can go to Drug Bank. So let's go to PubChem. just put chem in your folder. So now we want to download. Let's download all the 3 drugs before we start docking them. Okay, let's just do one first. So let's download MLN. I think it's MLN4. That's the name of the standard drug we are looking at. 4760. Can you see it here? MLN is a known inhibitor of ACE2. If you don't want to use MLN for your own study, all you need to do is go online, search for FDA approved inhibitor of ACE2 or any other enzyme you want to use as you are working with. FDA approved. For FDA to approve it, it means that drug is working well. That's before. So FDA approved inhibitor of HIV protease and hypertension. This drug, if you are targeting, if you are looking at developing drug towards hypertension, you understand the concept. So we are looking at this drug because it has been approved by FDA to be good for ACE2. So click on it. Can you see it now? This is the first one we have. So let's click on it. So can you see the compound, the chemistry name? Can you see the structure, the 3D and the 2D structure? This is the chemical formula, right? This is L-histidine derivative, that is L-histidine, which is hydrogen. Every information you can think of is already on this. We've gone through this one in the past. So, and anytime you want to do molecular docking, you don't work with 2D, you work with 3D. And I told you the last time that if let's assume there is no 3D, you can download the 2D and you use Avogadro to convert it to 3D. And I want to believe you guys already have Avogadro installed, right? On your laptop.
Yes,
I want to believe you guys already, yeah. So that's fine. So once you do this now, once just click on 2D, it will take us there. This is the 3D structure, right? And then click on download. Once you click on download, we are downloading the SDF. Click save. It will automatically save. Are you getting it? It automatically save on then go to folder. Let's go and copy it into our folder. Ctrl X. So go to lib folder. So because we are looking at different drugs, I want to have different folder inside this page for each of them. MLN. So let's copy it in MLN, right? So that's ML, that's it. So we have that. So that's MLN. Okay. So let's go and download. Okay, let's do molecular docking with MLN first. Now, once you already download MLN, the next thing is to go and open that protein on Chimera. And how do you do that? Go to File, Open, and go to your folder. Wherever you have it, we have it in lib MLN, right? And this is the structure. Double click on it. You see the structure of MLN here? You guys see it? You see it, right?
Yes, sir.
Okay. So that's the structure of MLN. So that's the structure. Okay. Can you please guys give me just 1 second? Can I? Just give me 1 second, please. Okay. Yeah. Okay. So now, okay. So now we have the structure of ACE2 now, right? So we have the structure of the MLN. We've downloaded MLN. How do we prepare MLN? When you want to prepare your drug, the drug now, you have to, for receptor, we remove hydrogen. For drug, we are going to add hydrogen. So we add hydrogen and we add charges. Those are the two things we do when it comes to preparation of ligand. Add hydrogen, add charges. So how do we add hydrogen and charges? Just come to tools. Can you see tools? Come to structure editing. Structure editing. And the first thing you see there is add hydrogen. Can you see? Add H. Click on it. It will bring you this box. Leave everything as it is. Click OK. If you look at it, it says what? It says hydrogens added. We've added hydrogen. The next thing is to add charges. Go back to tools again. Structural editing. Add charge. Click on it. When you are dealing with small molecule, make sure you change this to amber phosphate 14SB. By the time you download it, may not be in this format in your camera. So change it to amber F14SB. Let it be on gas charge and click OK. To bring you this box, and the next charge of this structure is +1. For advanced work, it is good to take note of the charges for each drug you are working on. Please, you take note of it. It is +1 for MLN. It may be something for another drug. So click OK, and it's going to add charges to it now. And when it's add charges, there is likely for this structure to adjust itself. So once charges has been added, we have prepared, we are done with the preparation of the ligand. So the next thing is to click on to save. Now, when you want to save, you save as mol2. You save your ligand as mol2, and you save your receptor as PDB. So the ligand, the drug is the ligand, you save it as mol2, click on mol2, and mol2 will take you to now go to, so I'm already in MLN, right? Let me give it what name, let's give it name. Let's give it MLN, right? MLN, leave everything as it is, and click save. Did you get it now? Click save. So we have successfully prepared our ligand, and we have successfully prepared our receptor. Are we fine? So once we have done that, the next thing is to do docking of the enzyme and the ligand together. So that's the next thing now we are going to do. So that is the next thing we are doing now. We've successfully prepared the ligand and the receptor. We have successfully done that. So please, you guys, give me like 5 minutes. So let's give me like let me just a bottle of water. So give me a 5-minute break. And you can also go on 5 minutes. You just take a walk. You get my point. So let me just pause this. Okay. So we've successfully prepared our ligand and our receptor, right? And we tell what is next now is for us to do our molecular docking. So we have we already downloaded and prepared the enzyme, downloaded and prepared the drug, and now it's for us to do the docking, which is, and I want you guys to take note of that. Straightforward. So now, for you to do your molecular docking now, let's go and bring our enzyme, the workstation. So our enzyme, the prepared one, right? Then once you have your enzyme, the next thing is to go and bring your drug also. The two of them must come on this space now. So let's go to this MLN, the prepared one. So can you see? So now we want to dock this drug at the active site. Don't forget, this is the active site of our protein, right? We want to dock this, we want to dock it here. So now, if we have to create this, for us to do the docking, let's go to tools. Go to tools, go to surface/binding analysis, and you come to Autodock vina. Autodock vina. I believe, let me go again. Use, then structure/binding analysis, then Autodock vina. And once you do that, this box is going to come out. You have this box. This box will come out, and this box is what we are going to use to give information to the software. Now, the first thing to do in this, you have to fill this box. And the first thing is to set what is called set an output. And now this an output means which folder do you want your docking document to go. So click on browse. So I want to save this in lib, right? Sorry, please. I will quickly press something. Okay. So I want it to save in lib, and I want it to save in MLN. Because when you have different drugs that you are looking at, you create folder for each of them, so that in the future, when you want to use it, you won't have results mixed together. So I'm saving this in MLN. By the time we are doing for Telenor, we also have a folder for Telenor we are going to save. So I want everything I'm going to do to be in this MLN folder, because we are docking MLN drug and H2. So let me give you, so if you ask me for a file name, let me give you H2MLN. You can give it any name, then you click on set output. So we already set our output now. Now, the receptor, the name of our receptor is 2.pdb, right? Is that correct? Hello?
Yes, sir. Yes, Dr. Ego.
Okay. The ligand is not PEP. So you click on it, then this is the ligand. This is the number of the ligand. Remember, it's not saved in MLN, so you click on. So we already indicate the receptor and the ligand. That's fine. Now, our interest is for this drug to bind at the active site of this protein, isn't it? Not to bind here, not to bind here, not to bind here, but at the active site of the protein. So if that is the case, you have to create a grid box around this place to tell the computer that no, you only want it to bind at the active site, not any other place. So we do what is called a grid box sizing. Now, the grid box sizing, this is where we're going to put it, but we don't have idea of what dimension it is. We are going to set it. What I advise people is just put 12 in all this thing. Just put 12. You can put any number. I just put 12. And you see, as soon as I click outside, this box comes out, right? Can we see this box? Now, once this box is out, let's go to, now, if I should move, I can't move this box now. If I should move this out, because I need to move this box to come at the active site of this protein. If I want to move it now, it's not going to move. Everything is going to move. How do I move the box alone? Go here. So click on resize and change this to button 1. You check the resize button 1. So if I should click on this now, I should be able to move this. Can you see? I can make it bigger, I can make it smaller, I can even move it around anywhere. But because this is where I want it to be, you bring it here, and I want it to cover the whole active site of the enzyme. So you can make it, just make it big at the active site of the enzyme. So, but for it has to cover the whole thing. For me to be able to know, I have to adjust, move it around. So I have to go back and move the whole thing. Let me just click on this so that I can move the whole thing. Can you see this? So that means it's too long here. And I need to make it to white. I can accommodate because these are the residues. This is the point that the enzyme, the active site of the enzyme. So let's reduce this. This one is too long, and let's make this one white. White rather. So go to this. I hope you guys won't have challenges in this place, because this is the point that many people used to have challenge to set the grid box. So let's go back so that for me to be, we have to move the whole thing so that we can know if. Do you see this? Can you see that this box actually cover the whole active site? Yes or no?
Yes.
So we have the box already covering the whole active site. Let me see. Okay, that's fine. So now, by the time we are doing the docking, the drug will not bind here, or anywhere. It will only bind within the box. And the box already, so when you are making this box, you must make sure your box is as precise as possible. Let it be covering the active site so that you are not docking your drug at another place. Now, another thing I want you to take note of, let's assume we want to do this for 10 compounds. Are we going to be setting these V box every time? You don't need to do. All you need to do is take note of these values. Think of these values. Think of the values. Think of the values that you are doing. Let's look at. So let's write it down. Can you write this down to me in a place? The center is 40.11. Write it as they are. Are you done?
Yeah, I'm writing it right now, sir.
Okay, please write it down. Okay. Before I switch, just write it down.
I took a picture of it, so it's fine. I'll write it now.
Okay, so you take a picture, that's fine.
Yes, sir.
So next time, if you want to do this for now, when you want to do for Telenor, or metformin, or any other drugs, you don't need to set this box again. Just input these values. It is the same protein, this same H2, the box will automatically be exactly where it is. Because if you are setting this again for Telenor, there is tendency that this box will not be exact as it is now. And that's going to be like a bias for your protein. That you are not docking the three drugs exactly at the same point. There's going to be a bias. So you take note of this. And apart from that, when you are writing your publication, your journals, for publishing, you have to give this information in your journal too. So those are the reasons why you take note of this. So the next thing to do is most of these values, put everything true first. Don't touch this. Are you getting me? Ligand, let it be true. Don't touch it. All this, I'm just showing you don't touch advanced settings. Leave all of them. Don't touch it. Don't touch it. Executable location. Yes. This is the point I want you to take note of. There are two ways you can use this software for molecular docking. You can use the internet. Anytime you are on the internet, you use it. But when you are not on the internet, how do you use it? If you want to use it when there is no internet, you check this box, local. This local box must be checked. Are you getting me? This local box must be checked. It's checked already for me. Are you getting me? Then you have to go and set. See, this docking you want to do, you want it to be in. You set it to MLN. It's in MLN, right? Okay, that's fine. Now, the executable language, the Python language, for this docking to take place is in a folder called vina. The folder is called, because the people that develop this program, they call it Autodock vina. So the executable programming language that you need for this docking to take place is in a vina folder. And for you to do that, you have to download the vina folder online. Are you getting me? And now in this case, I already have the vina folder. I'm going to put it on a Google Drive for everybody to download. Just give me 1 second. Just 1 second, please. So this vina folder, okay, you can let me show you. You can let's go and download Autodock vina. You just go to online and look for Autodock. Just type Autodock vina. Yeah, Autodock vina. This is it. Autodock vina. So scroll down. We should be able to get the vina file. Okay, download. Okay, so come, let me, no, that should be, okay, I know what I'm going to do. Okay, let's go back. Let's look for, let's look for vina, just type vina folder. Yeah, I think it should be in that. It should be in that vina. I should be able to get it in that vina. I'm coming. Let's. You need to get it most of the time on this. Okay, if I'm not getting it, I'm going to share it. I'm going to put it on the Google link, on the Google link, where you can download it. That's what I'm going to do. I should be able to get it here. Let me go to MDA. I'm coming. Just give me. No. Let's check Autodock. I want to see if you guys can do it yourself. You can just. Oh, what's wrong with this? You know, this know what I'm going to do. Okay. What I will send it to you guys so that you can install. You can have it. I will send it to you. I don't know why I can't see it here. So let me go to one of. Yes. So, okay. Okay. Yes. So this is the vina folder. Anytime you are doing docking, because you don't want to be using the internet, so that you'll be able to do your molecular docking using your local laptop without internet. Let's copy this vina and you put it in the folder. So I'm putting it in the lib folder. It must be in the folder you are using for molecular docking. You can see I already copied it here. So I'm going to put this on my Google Drive, and I will send you the link so that you guys can download it. Okay, that's fine. Let's go back to the Chimera. Let's go back to Chimera. Okay. So once you have done that and you click on this, so you click on browse so that we can go and choose the vina folder. I guess you can go to the vina folder for you to do molecular docking. So this is the vina. You click on the vina folder and you click on the vina.exe. That is the executable file that is needed for this study for molecular docking. You can see, just go to your folder, click on vina.exe, you click open. Once you have done that, we are completely done with anything that has to do with molecular docking. What you need to do is to click OK for the software to start docking. Any question before I click OK? Any question from anybody?
No questions.
Okay, so let's click OK and look at this place is running. It takes like few seconds, a few minutes. So it's running already, and it's going to give us the docking score. It will dock the drug at the active site of this site, and it's going to give us the docking score of this particular drug MLN to ACE2 specifically. So let's do that. Let's do that. So it's still running. Just give it, I think like maybe like 2 minutes, depending on the structure of the and the structure of the we are working with. Okay. So do we have any question? So that's fine. So while we are waiting for that to be done, let's quickly go back and download Telanor. I'm going to do for Telanor again, but you guys on your own will do for metformin, and you send me your docking score. Let's search for Telanor. Let's see. So this is, can see I'm on okay. Click on Telanor and click on we want 3D. Don't forget 3D, and to download. Save SDF, right? And let's copy that to the folder. Let's come to X. So let's go to the lib. So we want to save it in the, let's create a folder for Telanor. So let's call it PYL. So let's paste it there. Control S. Wow, that's fine. So let's go. I think the result is ready. Yes, the result of this is ready. Can you see this? Hello.
Yes.
Yes, we see it.
Can you see the, so this software is going to give you the best 5 possible ways that this drug can bind at the active site of this enzyme. The first one gives us minus, what is the score? Minus 8.6, right? Can you see that?
Yes, sir.
If I should click on the next one, minus 8.4, if you see that the orientation of this enzyme, of the drug rather, is going to change. Let me look, just watch this, and I'm clicking on minus 4, minus 8.4. Can you see? So if it's using this orientation, is minus 4.8, minus 8.4. Another one is minus 8. At 8 is this. So the one with higher negative value is the one that we go with most of the time, and that is minus 8.6. So please, everybody, take note of this. So our standard drug now that we are using as reference is giving us minus 8.6. So let's see what Telanor and metformin is going to give us now. So now let me teach you. So take note of this value, right? And don't forget the first one is what? The mode is 3.1, right? You have to go and save this. You have to go and save this because you will use it for, you can use it for other studies, other things that will give us information on whether this thing can work on this. Let's go to save this, go to file. Don't forget, because the protein and drug are concerned, we are going to save it as PDB. Now, so let's give it a name. Let's give it, let's call it, it's not, because we want to save the drug and the ligand and the protein together. It's going to be a complex. Let's call it final complex FCMLN is 2. So it is a final complex of MLN and is 2. Now, please, I want you to take attention to this. This is very tricky. Now, to save this now, to save the drug and the active site of the enzyme together, you click, everything here is highlighted. We don't need all of them. Just click on your receptor, your enzyme, the first one, right? And the best model that we want is the first one, 3.1, right? The best one that has minus 8.6. And for you to be able to just press Control on your laptop and click on this. Because if you do not press Control, when you click on this one will not be selected. So can you see, we select the receptor and the model that we want, the best one, which is 3.1. If the best one that you want to take is 3.2, it is that one you are going to pick together with the receptor. Did you get that? Hello?
Yes, sir.
Okay, that's great. Now, once that is done, make sure that this box, they are not ticked. But this third one is ticked. Save relative to the receptor. This is the receptor. Is there. Can you see? That one is 0.1. Now, save multiple model in there are two options here: multiple file, single file. If you save it as multiple file, that means it's going to save the ligand separate, the receptor separate. No, we want it to be saved together. So you click save as single file, as one single file, and that is the IP36. And once that is done, and you already want it to save in MLN, just click save. It is saved. Let me close it. Close. Let's go and let me open it for you before we go to Telano, to MLN. Can you see? Can you see FC MLN PDB? Can you see? The MLN is already, you see the two, this is, can you see the drug? Let me highlight the drug for you to see. You see now? That's brilliant. Tomorrow, next week, I'm going to, you will see that this thing that you are seeing is not just hanging there. It is interacting with amino acid on this protein. We are going to see that next week. Let's go. Now we've downloaded Telano, right? Let's go and prepare Telano. Let's prepare that. Let's go back. Now let's go to the process again. So this is Telano, right? And I said, if you want to prepare your drug, you add energy, you add charges and what? And hydrogen. And to do that, you go to file, tools. So now I'm going to show you how you can do the two together. You know, the last time we first of all did add hydrogen and add charges separately, but you can do the two together at once. And how do you do that? Just come to minimize structure. When you click on minimize structure, it will add hydrogen. It will also add charges. Let's click on it. It will ask you, put everything as it is, click on minimize, leave everything as it is, click on OK. The charge is FSB, Amber FF14SB, fine. Okay. You can see now, the next charge for Telano is what? Because it's 0, not 1. You know, MLN is 1 plus 1. This one is 0 now. So, okay. So we have, if we've prepared the ligand now. So once we've done that, let's save. You save your ligand as what? Mol2. So this is Telano. Let's give it TYL, right? So we are saving it in Telano. So save. So once we have saved that, so let's dock this to H2. Let's go back and open our, let's bring the two blocks, the H2, sorry, the H2 here. And let's bring our block to Telano. So now the next thing is to go to tools, right? Structure, surface analysis, then Autodock vina. Autodock vina, right? So I told you the first thing is to set your output file. Click on browse. So let's go and click. Sorry. Yeah. So now let's go to where we have Telano. We want to save it in, we want everything we are going to do to be in that. So let's give it H2 telanol, right? Then set output. Click on set output, is already okay. So now our acceptor is this. Do you know why this is not giving us PLY that we save it? Let's go back to, because that is the PDB number of this protein, of this, you see, 1983. And if you should go back there, that is why it's giving us 1983. Can you see? So it's not, you have to ligand, our ligand is the 1983. Now, instead of us struggling again, get a grid box now, instead of us doing that again, since we are using the same H2, so tell me, please call the number for us. Tell me.
Oh, sorry, I was muted. Yes, I was muted. Sorry. So it's going to be 40.1147.
1147.
Yes.
Okay.
And it's going to be 9.85.
633.
333. No, 633.
Okay. 6. Correct?
And yes, that's correct.
Okay.
And the next one is 21.76.
0.76.
15.
Okay. The next one.
And for the size, it's going to be 21.05.
Okay.
35.
Okay. The next one.
Next one is going to be 15.16. 49.
The next one.
Oh, 49. 49.
It's 49, not 19.
Yes, 49. All right. And the last one is going to be 17.9057.
Correct?
Yes, sir.
So can you see it's already set automatically? Instead of you disturbing yourself, once you do that, just take note of it. So the executable file should still be intact. So vina xex, yeah. So once that is done, so let's click OK. So let's see the score that telanol is going to give us now. Can you see the results? Hello?
Yes, Dr. We can see the results.
So the result that we have now is what?
It's negative 5.8. It's about the same as the last one.
Yes. So that's how, so if you have 100 compounds and you want to see which of them is going to work for H2, you don't need to stress yourself. Just do this. Do you get my point? Just do this. And that, so automatically, minus 5.8 to minus 8.6 is far. Definitely, telanol is not going to work against H2. Are you getting me now? Hello?
Yes.
Are you guys, I'm only hearing to me. Are you guys getting me? So if you have 100 compounds, you do this thing for all the 100 compounds. And when you do it for all the 100 compounds, and whichever drug, and when I say compound, I'm writing as drug, whichever drug give you something close or higher for your reference drug that is already in market, then these are the ones you are going to say, definitely this is likely to work against your target. Let me save this. So the first one is the one we are going with. So I told you save it as PDB. Now it's going to be final complex, but it's not going to be MLN, it's going to be PYL now. And don't forget this place again. I said choose your receptor number 1 and press Control on your laptop and choose the first drug model, the first one, 3.1. Everything should be fine. Single. Once you do this once, you don't need to stress yourself again on this. And we are saving it on telanol. See, You see that now? So this is what? This is saved. So can you, so now, that is what for metformin. I want to see result for everybody. For metformin on, send me your docking score for metformin. When you are sending the assignment I give to you on Thursday, you send the assignment, put the assignment in a Word document and send it to my email address on Thursday. And another thing you are going to put there is the docking score of metformin that you get from your own docking.